गुड मॉर्निंग। सभी का इस वीडियो लेक्चर में स्वागत। एमएससी फाइनल बॉटनी पेपर 5 के पिछले वीडियो में अपन ने फर्टिलाइजेशन के बारे में अध्ययन किया था। इसी कड़ी में इस वीडियो में जिस टॉपिक पर चर्चा करेंगे वह है एंडोस्पर्म या भ्रूणपोष। तो एंडोस्पर्म क्या है, इसके क्या टाइप्स है, फंक्शन, इसका साइटोलॉजी, ये सब अपन इस वीडियो लेक्चर के माध्यम से सीखने का प्रयास करेंगे। फर्स्ट ऑफ ऑल, एंडोस्पर्म। एंडोस्पर्म इज द मोस्ट कॉमन न्यूट्रिटिव टिश्यू फॉर द developing embryos in angiosperms. Functionally, it is comparable to the female gametophyte in gymnosperm but has a unique origin, whereas the female gametophyte in gymnosperms differentiates before fertilization and is haploid. The endosperm is the product of fertilization, is usually triploid. एंजियोस्पर्म के अंदर ट्रिप्लोइड होता है जबकि जिम्नोस्पर्म के अंदर ये हैप्लोइड होता है। After double fertilization, the egg is called zygote and the fusion product of polars and the second male gamete is the termed primary endosperm nucleus. The former develops into a organized embryo whereas the latter rise to an almost formless tissue, the endosperm. Endosperm is absent in the members of families Orchidaceae, Podostomaceae and Trapeaceae. Brachyria satigera and apomictic species is only example where endosperm has been reported to produce triploid embryos and seedlings. Development of endosperm. The primary endosperm nucleus is normally located directly below the egg cell and undergoes division immediately after its formation. During triple fusion, only the sperm nucleus fuses with the polar nuclei. Following fertilization, several changes occur in the fine structure of the central cell that are indicative of increased metabolic activity and organization of protein synthesis machinery for the differentiation of the primary endosperm cell. Depending upon its mode of development, 3 types of endosperms have been recognized. First, nuclear, second, cellular, and third, halobial endosperm. Out of the 288 families of angiosperms for which information was available at that time, 161 families show nuclear endosperm, 72 families cellular endosperm, and only 7 families have halobial endosperm. Cellular endosperm is largely restricted to dicotyledonous families in monocots only in Ericaceae and Lemnaceae. 17 families halobial endosperms, out of which 14 are monocotyledons. First, nuclear endosperm. In this type, the division of primary endosperm nucleus and few subsequent nuclear divisions are not accompanied by valve formation. This results in a condition where the central cell of the embryo sac has formed a few to several thousand nuclei freely suspended in its shape. Such a condition, endosperm may persist until it is consumed by the developing embryo. When later in this case, which is more common, the valve formation is mostly centripetal, that is from the periphery toward the center. The degree of cellularization varies a great deal. Mostly the endosperm becomes completely cellular, but in fasciolus cellularization occurs only around the embryo. In this slide, we see nuclear endosperm in Eclephi indica. Development of endosperm in coconut deserves special nutrition. The primary endosperm nucleus undergoes a number of pre-nuclear divisions. When the fruit is about 50 mm long, the embryo sac gets filled with a clear fluid in which float numerous nuclei of various sizes. At a later stage, about 100 mm long fruit, the suspension shows, in addition to pre-nuclei, several cells, each enclosing a variable number of nuclei. Gradually, these cells and pre-nuclei start setting at the proper periphery of the cavity, and layers of the cellular endosperms starts appearing. This forms the coconut meat. The quantity of the cellular endosperm increases further by division of the cells. In mature coconuts, the liquid endosperms become milky and it does not contain prenuclei or cells. Second is cellular endosperm. The cellular endosperm is characterized by the absence of prenuclear stage, the division of primary endosperm nucleus, and a few subsequent nuclear divisions are followed regularly by valve formation. The occurrence of Historia is common feature of this type of endosperm. It is more varied than that the nuclear endosperm. The Historia may be micropylar or chelazal. Occasionally, both types of Historia are present in the same plant. Deviation from the general pattern of endosperm development in acanthaceae is shown by Bleipheris and Thunbergia. The linear three-celled stage present in all other members of the family does not occur in these plants. They also lack the chelazal Historium. After the division of the primary endosperm nucleus, a micropylar chamber and a chelazal chamber are formed. The former grows into an Historium, whereas the latter gives rise to the endosperm proper. In Thunbergia, the micropylar and Historium is a balanced branched sinusotic structure. In this slide, we see cellular endosperm in Drymis winteri. Clusia notoniana shows an interesting pattern of Historium development. Both chelazal and micropylar Historia occurs in this plant. The development of endosperm in the Lorenthaceae is unique. There being no true ovule, all the embryo sac in the ovary lie close to each other. After fertilization, the primary endosperm nucleus moves to the basal part of the embryo sac where it divides. During their development, the endosperms of all the embryo sacs in an ovary fuse to form a composite endosperm. Halobial endosperm. This type of endosperm is restricted largely to the monocotyledons. The primary endosperm nucleus moves to chelazal end of the embryo sac where it's divided, forming a large micropylar chamber and a small chelazal chamber. In micropylar chamber, prenuclear divisions and cell formation, if any, start at a much later stage. In the chelazal chamber, the nucleus either remains undivided or divides only a few times. In this slide, we see halobial endosperm in Asphodelus. Cellularization of endosperm. Ultrastructural studies have provided useful information on the cellularization of the nuclear endosperm. Reports on the initial stages of valve formation in wheat are at variance. Morrison and O'Brien, 1976, reported that the formation of the first layer of endosperm cells does not involve phragmoplast. According to them, the process of cellularization starts with the development of centripetally growing walls from the central cell wall. The preends of these anticlinal walls branch and the branches from the adjacent walls fuse on the side nearest to the large central vacuole. Then next topic, cytology of endosperm. What is cytology? In majority of plants, the endosperm which is triploid because it is derived from the fusion product of three haploid nuclei, one from male gametophyte and two from the female gametophyte. Whereas the number of nuclei contributed by the male gametophyte in the formation of endosperm is constant throughout the angiosperms, the number of nuclei contributed by the female gametophyte varies with the type of embryo sac. The endosperm tissue is well known for a high degree of polypolarization of its cells during development. Average 1965 studied endosperm cytology in many flowering plants. The highest ploidy, however, has been reported in the Arum maculatum, where the nucleus becomes 24,576 n means ploidy. Endomitosis and nuclear fusion are some of the methods of polyploidization of endosperm cells. The occurrence of various mitotic irregularities such as chromosome bridges, lagging chromosomes, spontaneous breakage of the chromosomes, and fragmentation of the nuclei is quite common in the endosperm tissue. Size of the nucleus and the number of nucleoli per nucleus also exhibit great variation. Exception to the rule in Dendrophthalcata, where the endosperm cells show remarkable stability in their chromosome number, all the cells of mature endosperm are triploid. The endosperm is usually non-colorless in crinum. During seed development, the seed coat as well as fruit wall are absorbed and the endosperm is exposed to sunlight. Consequently, it becomes green. Chlorophyllous endosperms also occurs in Metiola, Raphanus and Viscum. Last, functions of endosperm. In plants with albuminous seeds, the endosperm reserves support early seedling growth. However, a more important contribution of endosperm in the nutrition of embryo during its early stage of development from zygote to globular embryo. This conclusion is based on the following observation. First, at the time of fertilization, the embryo sac has very little nutritive material. After the endosperm advances in development, it stores enough food substance to ensure an adequate supply for the developing embryo. In the majority of angiosperms, the zygote divides only after endosperm has reached a reasonable stage of development. Generally, the embryo grows only when the endosperm develops properly. In the absence of endosperm, or like in Orchidaceae, Podostomaceae, and Trapezae, special provisions exist to ensure the nutrition of developing embryo. During its growth, the embryo depletes the surrounding cells of the endosperm of their contents. In some creels, several layers of endosperm at the base differentiate into transfer cells with extensive wall ingrowths. This specialized tissue helps in transport of metabolites from the maternal tissue into the endosperm. Apart from being a nutritive tissue, endosperms also regulates the precise mode of embryo development. Removed from the influence of endosperms, very young embryos often fail to mature even in the richest artificial nutrient media. Moreover, the isolated older embryos often skip the normal stages of embryogeny and show precious germination in culture. So, in this topic, we studied endosperm, what is endosperm, its types, then cytology, and then functions of endosperm. Thanks to all in this video lecture.